Translation in prokaryotes. There are three stages in translation: initiation, elongation, and termination. Firstly, for initiation to occur, two ribosomal subunits, the small 30S and large 50S subunits, are required. Both initiation factors, IF1 and IF3, bind to 30S subunit. IF1 prevents tRNA from binding to small 30S subunit, and IF3 binds to the free 30S subunit, preventing large 50S subunit from binding to form an inactive ribosome. The third initiation factor, IF2, is a GTPase that binds GTP. IF2, which carries a molecule of GTP and the charged initiator tRNA, bind to a free 30S subunit. After all these have been bound, the 30S initiation complex is formed. The AUG or start codon is recognized by a special tRNA, tRNA FMET, carrying a modified methionine, N-formylmethionine, FMET. The initiator tRNA FMET forms base pairs with the AUG initiation codon. The 30S ribosomal subunit binds at the Shine-Dalgarno sequence or ribosome binding site. The Shine-Dalgarno sequence forms transient base pairs with part of 3' end of the 16S rRNA component of the 30S ribosomal subunit. This allows binding of 30S subunit to mRNA. The 30S subunit then migrate downstream until it reaches the AUG initiation codon. The 50S subunit then binds to the 30S initiation complex. The GTP carried by IF2 is hydrolyzed and all the initiation factors along with GDP and pi are released, resulting in the formation of the 70S initiation complex. When the 70S initiation complex is formed, there are three sites for tRNA binding called the A site, amino acyl, the P site, peptidal, and the E site, exit. The P site is now currently occupied by initiator tRNA FMET that is bound to the AUG initiator codon, and the A site is free for the next tRNA to bind to. The E site is currently free as it is where the uncharged tRNA is to be released. Three elongation factors, EFTU, EFTS, EFG, which bind GTP, are required. Elongation occurs when the elongation factor, EFTU, guides the tRNA with the correct anticodon to enter the A site and form base pairs with the second codon. Once the correct tRNA is added, the GTP is hydrolyzed and EFTU-GDP is formed. EFTS and GTP reactivates EFTU-GDP for later rounds of elongation. Currently, both the P and A site each contain a tRNA that carries an amino acid that is in close proximity. Peptidal transfer activity catalyzed by peptidyltransferase of the 50S subunit occurs, causing the formation of peptide bonds between FMET and the second amino acid. tRNA decylase breaks the FMET tRNA covalent link, and the A site now has the tRNA that is attached to the newly formed dipeptide, whereas the P site now has the uncharged tRNA. Translocation occurs whereby the ribosome moves 3 nucleotides along the mRNA in a 5' to 3' direction, causing the uncharged tRNA to move from the P site to the E site, and the tRNA containing the dipeptide to move from the A site to the P site. The elongation cycle repeats. The net result of one turn of this cycle is that the polypeptide has grown by one amino acid residue and the ribosome has moved along the mRNA by 3 nucleotide residues. The uncharged tRNA is now removed and the A site is now free for another tRNA to enter. 3 release factors RF1, RF2, RF3 are required for termination. Termination of translation occurs when one of the stop codons, UAA, UAG, or UGA, enters the A site of the ribosome. No tRNAs correspond to those sequences, so no tRNA is bound during termination. One of the 2 release factors, RF1 or RF2, enters A site, helped by RF3, causing peptidyltransferase to join the polypeptide to a water molecule, releasing polypeptide from final tRNA. The ribosome releases the polypeptide, and once the protein is released, ribosome recycling factor RRF and EFG work to release the uncharged tRNA from the P site. The ribosome then dissociates. It is stimulated by the binding of the IF1 or IF3. The 50S subunit leaves and the 30S subunit may either dissociate from the mRNA or move to the next start codon.